All right, hey guys, what's going on? So today I want to talk about a little bit the methods that go into the type of area that I'm studying. And I would love to hear your guys' inputs on and questions that go into this, because a lot of what we're going to be talking about is how physics meets the road with biology. This is straight up biophysics. And I want to talk about this because, just in brief, maybe just 10, 15 minutes, I want to talk about this because it's all fun and games when you're talking about the theory. Some people take this view, right? Some people say, it's all fun and games to theorize about the quantum physics of protein dynamics, or the statistical mechanics that goes into protein folding or something like that. But when push comes to shove, what people want to see is the evidence. What people want to see is, how are you testing your theories in practice? And this is where I want to talk about a little bit. So what I want to talk about, I've already pre-written CryoEM on this. I want to talk about, just in brief, CryoEM and some electrophysiology. And I want to talk about these within the context of the receptors, so let's get right into it. CryoEM stands for cryogenic electron microscopy, and EPhys is what some people will call it. It's called, whatever in here, electrophysiology. But I want to talk about how these methods work, just in brief. I will probably cover them deeper as we go through this lecture series. Now, CryoEM, essentially what you're doing is you have an electron gun, basically. Cryogenic electron, right? This is your electron microscopy. You have some electron gun. And this electron gun will fire electrons at super high speeds. So you use these, these coils, coils or these wires, to focus and speed up electrons that go really, really fast. And then you have your sample. Right, so your sample is on some grid. Right? And when the electron comes in contact with your sample, it then produces a pattern, right? I don't want to say right. If you guys don't know this, then I can't say that. But anyways, it then produces it projects some sort of pattern, right? And how does it do this? Well, there's a lot of quantum physics that goes into this, right? Because one single electron has an associated wave-like nature to it, right? And so if you are sending an electron towards a target, and if you can think of it as a wave traveling towards the target, you can sort of think of it as, when it comes in contact with the target, a ripple effect will happen, or sort of a rippling effect, if you will, occurs within the wave, and what you get is a sort of a specific wave thumbprint, if you will, of the orientation of the protein that it's bombarded with. If your protein is in a slightly different orientation, then you're going to get a different pattern within that probability wave. And that probability wave, so what you do is you freeze this sample right here, right? So I'll do this in blue. This sample here, you have tons and tons of sample, right? So this is a little grid that contains a lot of protein, right? Each protein has a slightly different orientation to it. And because each one has a slightly different orientation, you can take each one produces its own little unique thumbprint pattern within the projected wave that it came in contact with it. And you can take each one of those orientations, right? So you take each one of the orientations. So say one orientation looked like this, the other one looked like this, the other one looked like this, and you have, say, 100 or 200 orientations. You sum them all up in a three-dimensional way, and what you get is a three-dimensional protein, right? So it's my feeble attempt of drawing a three-dimensional structure. Right. And that's what's called a map, right? So this is a map of the protein. And that's how, there's a lot of mathematics that go into it. There's a lot of computational stuff that goes into this. There's a lot of physics, again, that you have to understand. And we'll go into the details on this later. This is essentially what CryoEM is, though. You use electrons and you throw them a frozen sample. That frozen sample consists of a lot of small particles in it that you're interested in studying. And then from those, you can take all the orientations, add them all up, and you get a map. So we're going to put CryoEM aside now, and we're going to talk a little bit about electrophysiology. So, but before we do that, this is a way of studying the structure of proteins. And this is important, right? This addresses the question of structure. Electrophysiology is going to address the question of function. So there's a bunch of different methods that go into studying studying the function of proteins, and electrophysiology is one of them. And essentially what you do is you take a cell. So I'm going to take a cell. Here's my cell. And here is my protein on said cell. I'll draw it in green. Say this is a channel of some sort, or we're going to say this is a glutamate receptor, right? So this is our glutamate receptor, and what you do, this is very interesting. This is very, very interesting. You take a very fine needle. This is really not to scale it, but you take a really fine needle. I'm blowing this up like way zoomed up, if you will. So you take a really, really fine needle and you move that needle so that it comes in contact with the cell. Right? And what does this do is it selects a small patch on the membrane of the cell. And I'll just get rid of these guys. You're selecting a small patch on the cell, and then essentially what you do, again, I'm not going to get into the details right now, but essentially what you do is you apply any concentration of ligand that you want in here, right? I should have made those a different color, just not for us to get confused. Right, so you apply any concentration you want, and what you can then do is you have an electrode in here. I'll make the electrode dark blue. You have an electrode in here, and as each one of these comes in contact with your receptor, there's going to be a conductance of ions that pass through, and we all know that if there's a change in the concentration of ions that pass through, so right, you have ions also, so say the yellow are ions. So what I'll do is I'll make yellow ions. So you apply some concentration of glutamate, for example, then ions will pass through, and the change in the ions, the change in the concentration, will contribute to a change in the voltage because ions contain charge in them, and when ions contain charge in them, that means there's also an associated voltage with that charge. So with this conductance, comes a change in voltage. Okay? And so when there's a change of voltage, then this guy will measure that change in the voltage. And then, essentially, you could take this change in the voltage to a computer. This is hooked on to a computer. Right. And you have your computer, and the curve you get is going to look something like this. Okay, now what this means is that at this point here, the channel opened. So the channel opened at this point, and it reached a maximum, right? So then it reached a maximum here, reached a max voltage, okay? And then at this point, it starts to close, right? So the voltage starts to re-equilibrate, and there is a closing in the channel, right? So channel closes. And essentially, what you can do, so very, very interesting, what you can do is you could say, well, say I expose my channel to glutamate, and upon the exposure of the glutamate, I also add a partial agonist or any other chemical that you think might have an effect on this receptor. So if I add a partial agonist or some sort of antagonist or something like that, then this curve is actually going to change. Right? If you're doing your experiment carefully enough, the rate at which the channel closes, this is basically what you're measuring. They're measuring the rate at which the channel closes. And so you can add chemicals to your solution to speed it up or to slow it down. Right? You can add chemicals that render it completely open. Right? And we talked about this a little bit before. You can add a channel that renders it completely closed, or add a ligand or some chemical. And that is extremely important as well, right? Because that helps you determine what are the ligands and what are the chemicals that are good for these receptors, given that you define the term good, or what are the chemicals that could potentially be harmful. And if we're talking about receptors that contribute to disease states like schizophrenia, Alzheimer's, Lou Gehrig's disease, or any sort of mental disease, anything really, this applies absolutely everywhere in the physiological domain. That's essentially pharmacology, right? So this is the basis of understanding pharmacology. And so that's what I want to talk about, right? So briefly mentioned CryoEM. This addresses the structure question. What do these proteins look like? And electrophysiology addresses the function question, right? So function. Because there's this mantra in structural biology that with structure comes function. You look at a desk, for example, you look at a table, you can automatically infer what its function might be. You might not be right, but you can think, well, there must be some function. If I could see something and it looks like it has some sort of structural coherence or structural pattern to it, then you might be able to infer, well, maybe it's supposed to do something like this, or maybe it's supposed to act like a lever or something like that. It has a function. You're inferring some sort of function given that there's a structure. What I want to talk about later, and this is something that I'm going to be getting into my research in a little bit later also, is going to be localization, right? So how can we understand the localization of receptors, particularly these glutamate receptors in the brain? Because it'll turn out that there are some types of glutamate receptors that are not just postsynaptic, but also presynaptic. And they have various different types of functions. My apologies for the dog barking. I will stop there just in case he starts barking again. There he goes. And I will see you guys in the next one. Bye.